Welcome to Inside Bioprocessing, a Thermo Fisher Scientific podcast discussing key trends and topics within the bioprocessing industry. In this episode, we'll be discussing the application of multi-omics analysis for cell culture media development and optimization. During biopharmaceutical production, developing a medium that can meet the unique nutrient requirements of the cells producing the therapeutic molecule is central to achieving optimal results, both in terms of cell growth as well as protein yield and quality. One emerging technique which could dramatically streamline this process is multi-omics analysis. To learn more about multi-omics and its growing role in bioprocessing, we're going to speak to three Thermo Fisher media development experts. Please join me in welcoming Senior Manager, R&D Multi-Omics, Paul Gouldy, Staff Scientist, Shenzhen Tu, and Senior Global Product Manager, Bioprocessing, Femi Egbebi. So it'd be great, Paul, for you to first start and explain why is detailed analysis so important during the media optimization process?
Certainly, anytime you are developing a medium or feed, one of the things that you need to account for is how each metabolite within that medium or feed is going to be utilized. And so to do that, we need to get very detailed analysis of the bulk metabolites that a cell line would use, such as amino acids, carbohydrates, the vitamins that would be utilized. And if you think about it in terms of if you were trying to have a specific diet for, say, running a marathon or gaining weight for a weightlifter or something along those lines, the nutrition that that person would be taking into their bodies would be incredibly important. They would want to eliminate certain aspects of that or increase certain aspects of metabolites that they would bring into their body. The same is true for a cell culture process. The cell lines have specific needs, and in order to determine what those needs are, we need to have a very detailed analysis of, what is going into those cells in order to get what we want out of those cells to be accurate and complete.
For spent media analysis, how long has this been the industry standard for media optimization? How exactly does it work, and what would you say are some of its limitations?
Sure. In terms of spent media analysis, I would say it's been the industry standard at least for the last 3 decades, I would say. Um, Really taking hold in the 90s is when uh, industry started looking in depth at what was occurring inside the medium. Up until that point, a lot of work was done around basically dose curves where we knew certain metabolites were needed, right? All cells need methionine, so you can do a dose curve to determine, how much methionine to add to the formulation, and that dose response would give you, a basic, understanding of what to put into the formulation. But as better technology came along to be able to look at spent media, um, it became more of a push to look at wholesale amino acids, carbohydrates, those things. And I would say that again began in the 90s with the goal of removing materials such as hydrolysates, and that's where the work really started. Was um, Groups were trying to determine what were hydrolysates bringing to the formulation that were causing it to perform well. When you use something like yeast extract or P hydrolysate, what exactly is contributing to the performance of the cell line? And the goal was really to remove those things because they were relatively undefined. We didn't have a clear understanding of exactly what was in it. And that always brings a certain amount of risk, particularly to the final product. It also makes downstream purification of those things very difficult. So, about the 90s, we started to want to remove these hydrolysates, start to be able to bring in, chemically defined formulations. One of our earliest chemically defined formulations of CDCHO was an immediate hit because so many groups were looking to remove those materials from it. And so that's where spent media work started. And how it exactly works is you're going to take sample over a period of a cell culture process. Generally, most samples plans will start with a day 0 sample, and then something that's like 3, 5, 7, day 12, day 14, something along those, so you can see across time what is happening to all the metabolites within the formulation. So you'll take your sample, you'll spin out your cells, try to clean it up a little bit to get it so that you can actually run it across the LCs or the liquid chromatography in order to be able to see what is actually in the formulations. The limitations really are about you're looking at just the spent media. You're not actually looking at what the cells are doing with those metabolites. So really, all you're seeing is a limited amount of components, maybe 30 to 50 components in total. And you're seeing that, okay, well, cysteine is running out, but you're not seeing what that cysteine is being used for by the cell. You just know that it's running out. And you could add more in hopes, and that's what many people did for the longest time. They just added more cysteine. But sooner or later, you're going to get to a solubility limit where you can't add any more. And that's where you need to start then asking questions like, well, why is the cell needing this much cysteine? What is it doing with it? And is there something else that we can bring to the table other than cysteine to satisfy that need? Spent media really doesn't give you those answers. It just lets you know that it's running out in essence. Um, Additionally, it'll give you information about what is accumulating. Alanine is accumulating to very large concentrations. Well, why? What's driving that? You don't really know. You just know that it is accumulating. So there's only so much you can take out if the cell is creating alanine, right? Obviously, you can't just remove it. And that can create some problems later on down to in the cell culture process where, that can lead to increasing levels of lactate, increasing levels of ammonia, which then hurt the cell culture process ultimately. So that's really the limitation. It's just telling you what's going up and what's going down. It's kind of a shadow of what's actually happening. It's not really telling you what is happening.
And I guess this transition is perfect because I would say that that need plus the better technology coming along has brought in multi-omics. So actually, Shenzhen, would you be able to explain what multi-omics is, as well as the different types and where they fit within process development?
Yeah, sure. So speaking of Metabolomics, I would like to talk about the first omics, like genomics, right? So which is like the study of all genes and their interactions, like in a given species. So here, like one scientist I think should be mentioned is like Frank Sanger, right? The father of genomics. He is the only one won like two Nobel Prizes in chemistry. So after genomics, like scientists follow the same way, right, to study all the proteins or in like cells. So we call it proteomics. The study of all like mRNA in cells we call like a transcriptomics, and those like other like lipidomics, so on and on. Also have some other like I would say partial omics, like glycomics to study like glycans, and phosphoproteomics to study like phosphoproteins, and also such as other like kinomics to study all like kinase in a cells, right? So actually, Metabolomics like is like to like combine and integrate two or more omics data sets to investigate like biological pathways like in a more comprehensive manner. So give us a full image of the cells or tissue or our species. So in our like CHO cell based like expression system, like all of like genomics, transcriptomics, proteomics, and metabolomics are key omics. But like usually like from like a genotype to phenotype, proteomics and metabolomics are like more close to phenotype. So we think like proteomics and metabolomics are like key omics within like process development workflows.
What are different types of technology that can be leveraged when conducting this sort of multi-omics analysis?
Yeah, so for like genomics, epigenomics, or transcriptomics. Right now, scientists usually using like a next generation sequencing, right? Because the first generation sequence definitely is like a Sanger's message, Sanger sequencing. So right now is the next generation sequencing, like those RNA-seq can cover all of those RNAs, long read, so can give a comprehensive understanding of our transcriptomics. And for proteomics, metabolomics, or lipidomics, scientists usually use gas chromatography, mass spec, or high performance liquid chromatography, mass spec, it's like a GCMS or HPLC-MS to study proteomics and the metabolomics. And also another way, since we have different omics, right? We need to integrate them together. So we need lots of bioinformatics tools to perform data integration and data interpretation, and lead to our a final a DOE to optimize cell culture media.
And when it comes to using omics media optimization, so proteomics and metabolomics are clearly two valuable tools. How does each work and how do they contribute to media optimization?
Yeah, so I can go into that a little bit. A lot of times when I'm thinking about how proteomics and metabolomics work as tools to each other, I often think of kind of a roadmap in terms of what is happening inside the cells. And if you think about a cell as having a series of metabolic pathways in which metabolites will flow through, at either in terms of catabolism or metabolism, then you can think of those pathways as city streets, let's say. And if you have a proteomic view of what's occurring inside the cell, then you know which of those streets are actually available for you to go down and which aren't. Okay. You also have a sense of what's regulating those pathways. And going back to that analogy of city streets, um, where are the traffic cops basically staying, or where is there a traffic camera that's going to take a picture of you speeding through a pathway, right? So what is regulating those pathways and turning certain pathways on or turning certain pathways off? That all is going to be coming from the proteomics portion of it. The metabolomics gives us then that context of where are the metabolites actually going. So a pathway could be there, it could be available for the cells to use, but if the metabolites are not flowing through that pathway, then something is stopping that pathway from being used. And how does that change over time? So you really need both. One omic will only give you half of the story. It won't give you the full story. So that's what we're attempting to do within our group. We'll sample any given cell culture process for both a proteomics samples as well as metabolomic samples, run them on their individual mass spec units that will give us that data set. And then once we have identified the proteins and been able to quantify what's occurring and identify the metabolites and quantify what's occurring there, we'll then sync those two up and be able to get a model of what is actually happening in the cell. And then from that, we try to determine, well, what attributes of that are affecting titer or affecting growth or affecting a product quality attribute, and what are the influencers on those aspects that we really care about in terms of the production of the therapeutic. And from there, then we will design some experiments around that, around those pathways to try to get to that final formulation.
Shenzhen, would you be able to explain how process development scientists can take this information from multi-omic analysis and then turn it into actionable steps in order to optimize their media formulations?
Right now, I believe the information we got from metabolomics analysis is still the tip of the iceberg. I would like to say to separate into two main steps, right? The first step is like a help cell to survive, which means that we need to give cell what they like, right? So for example, like from like a Multi-Omics analysis of like the enzyme levels and the metabolic flux, which will kind of tell us the cell like for example easily induce like a glutamate into like a TCA cycle to generate energy. Then we will design maybe like add more glutamate into our cell culture media, right? Since like the cell will consume it and will like induce glutamate into TCA cycle, like generate more energy and improve like cell growth and improve protein synthesis, right? Like that. So first step, give what the cell like. So to help the cell to survive, improve the cell growth, improve the cell viability. The second step is to like we want to like push cells to change from survive to thrive, right? Which means like based on like our Metabolomics analysis, we will add some components to improve like some key cell processes, like such as like transcription, translation, or protein secretion, right? So it's like a process. Also, we can like add some components to like activate or like inhibit some key pathways. So we kind of wanted to add like more components to manipulate the cells to like produce more like therapeutic proteins. That's the second step, like that.
And Femi, do you have anything else to add?
I just want to emphasize what my colleagues they've mentioned about using multi-omics for media development. Just because you have the opportunity to really leverage intracellular information when you're making decisions about your process versus using spent media analysis, which really uses only data from the media. So you're essentially working with a lot more information to determine what's going on within your process, as well as it allows you to just get to the optimal process very much fast and better.
So now looking at some of the implications and the impact of multi-omics specifically for the biopharmaceuticals industry as a whole. Paul, are there any areas where technology development, such as within data science, could help advance multi-omics?
Yes, absolutely, there is. As you can imagine, when we have made this transition from a spent media approach, which is going to produce a certain amount of data and information for us to be able to use, to a multi-omics approach, that amount of data and the depth and of that data has increased exponentially for that. So to kind of give a sense, a spent media approach data set will probably give you maybe 100 to 200 megabytes of data. Relatively easy data to utilize simple programs to be able to analyze and determine, what your next step should be around that. A multi-omics data set can be upwards of 500 gigabytes in size. It's looking at 7,000 proteins, 1,000 metabolites, and it's looking at it across a time course and also in the context of the cells where those proteins actually exist in terms of the organelles or um, the structure in which they are related to each other. And all of that now gets to a point where using a simple program like Excel is just not going to cut it in terms of being able to do this type of analysis. You really need to go to a higher level of data analysis to do this. And so this is where data science can really come in. There's been a lot done around being able to get good identities of what these proteins are and quantification around it in terms of data science. And that's just, and the same is true for the metabolites, being able to identify metabolites, identify certain spectrums. But actually, the field is still somewhat new in terms of combining these very large data sets into usable digital models that can actually churn out and give suggestion as to what the next experiment will be. And those are areas that we are actually focused on very much, is being able to determine ways to utilize data science to do this advanced analysis and bring in the two omics together around that. And so that's where our focus is, and that's where we're building out our capabilities.
It's really interesting. Shenzhen, would you be able to touch on any other opportunities for multi-omics, both for media optimization as well as some other areas within bioprocessing development workflows?
The answer is definitely yes. Metabolomics include all those techniques from genomics, transcriptomics, proteomics, metabolomics, and those metabolomics, lipidomics, right? All those omics methods. So it definitely can be used in all the upstream bioprocess uh, and cell line development, right? Also can use the to study transfection efficiency, right? So if we have different conditions shows the transfection efficiency is different, we can use metabolomics to understand what's going on and tell you what's really important. Let's manipulate those pathways or at a more activate or inhibitors to improve our transfection efficiency, so kind of thing. Also can do some improvement for single cell cloning. We can help improve the single cell cloning efficiency, right? And and also for a downstream workflow for manufacturing, so those thing I think spec based method also can perform those quality control of our products or many of others HCP analysis, so to reduce the risk of our products, so kind of thing. So I will say metabolomics can identify differentially express genes, proteins, metabolites, lipids, so among different conditions. So it can be applied in each step of our bioprocessing development workflows.
So just to finish up, why is multi-omics so important to support the future of the biopharmaceuticals industry?
As we are developing new molecules, they're becoming increasingly more complex in terms of biotherapeutics. In addition to that, we now have the field of gene therapy and cellular therapy are beginning to become more and more important in terms of creating new therapies. And as a result of this, the need to have very robust processes is going to become more and more evident. If we create a new molecule that is going to cure, say, a cancer, particularly, say, a cancer for a child, and the cost of the production of that molecule results in a price tag that is so high that only the very rich could afford it, or it becomes to the point where it doesn't become financially possible for a pharmaceutical company to actually produce that molecule. That means it's not going to have the same effect, right? We're going to have many people who are not going to have that therapy available to them, and it'll also create a lot of strife and issues that we deal with all the time in terms of you know the socioeconomics behind the pharmaceutical industry. So, in order to create more robust processes that are going to produce these molecules in the constant quantities that we want, to the quality that we want, in an affordable way, so that it can be democratized across all people, then we're going to need to have a much more in-depth understanding of how these processes work and what levers we can pull to make them better. And Multi-Omics does that. Spent media is a fantastic process. The traditional approach has worked for many, many years, but we are very quickly getting to the point where the improvements that we can get from the traditional approaches simply aren't going to get us to where we need to be in order to enable some of these new technologies that we want to produce in biopharmaceutical industry. And so that's where I see multi-omics playing a critical role in giving us the understanding that we need to be able to get these therapies in a way that they are actually affordable and available to everybody rather than just a select few.
I think that's a great place to conclude this conversation, and I'd like to thank all of our guests for sharing their insights and expertise into this really exciting topic. I'd also like to thank you, our listeners. If you'd like to learn more about Thermo Fisher's Multi-Omics Media Development Service, please visit thermofisher.com/media development or reach out to a Thermo Fisher representative.